Start. Hello and welcome to Global IMMUNOTALKs and today's rendition. My name is Burkhard Becher, and on behalf of the organizers of Global IMMUNOTALK, it is my great pleasure to welcome you all, but in particular our guest today, and that is Sonia Garel. Sonia Garel is a bit of an odd bird today because she is a developmental neurobiologist rather than an immunologist, but she will be talking today about the role of a cell that we as immunologists claim to be part of our study focus, which is microglia, and specifically the role of microglia in early brain wiring. So we as the organizers felt she would be a wonderful addition to Global IMMUNOTALKs, and we're eager to hear her. Just a few words about Sonia. So after she obtained her PhD in developmental biology in Paris, she did a postdoc at UCSF with John Rubinstein, and then she came back to France and joined the Institute of Biology at the École Normale Supérieure, where she is heading the brain development and plasticity team since 2008. So Sonia's research focuses on mechanisms that control, on the one hand, the assembly of cortical circuits during development, but the particular interest is the role of microglia and the interactions with the immune system and environmental signals and the microbiome. And Sonia's work has specifically lately been super recognized, and she's been awarded a lot of, gotten several awards and honors. She's become a European Young Investigator Award. She received an ERC Consolidator Grant, the Antoine Lacasson Prize. She's a member of EMBO. The really big deal is something that a very few people can say, certainly under the age of 100, is that in 2020, Sonia was elected a professor at the Collège de France in the Neurobiology and Immunity Chair. So we're super thrilled here to have Sonia here today. And as it is our custom at Global IMMUNOTALKs, what we do is we ask our speakers a set of questions. And we asked Sonia, and I'll pass on to you right now, what is the advice that you would give to your 25 to 35-year-old self? Sonia, to you.
Thank you very much. So I'm absolutely thrilled to be here today, virtually. Good morning, good afternoon, good evening to everybody. Before thanking the organizers for putting together this fantastic series of IMMUNOTALKs, Global IMMUNOTALKs that I think are absolutely fantastic. And also for inviting such an odd bird as me as well. It's a real honor and pleasure. So what advice would I give myself? I think the advice I would give is not to follow too much advice. Because I think that if I had really listened to everything that I was told, I would never be a scientist. And I very much enjoy this work and the passion and the enthusiasm that it's bringing on a day-to-day life. And that I think that you need to be a bit crazy to do this, especially in these times. And so, yes, do not follow too much advice that people give you. Try to find your own path, what interests you most, your passion, your dreams, and just follow them and go for it. So I don't know if it's a good advice not to listen to any advice, but that's the one that I think I would have I've been happy to had a few years ago, especially when I was wondering what I would do, if I should stay in academics and pursue the work that I was eventually going to do.
Well, I'm glad you didn't listen to somebody else's advice. I'm glad you didn't, because it's been a pleasure to look at your work. And we've met several times. You're a wonderful person. So we're very happy to have you. This is good advice, much appreciated. And so without further ado, I'll pass on to you right now. If you could share your screen and you get started. Thanks, Sonia.
All right. Thank you very much. Yes, this is working. So again, I'm very happy and honored to be here and to tell you a bit about the brain. So today, I will be focusing on microglia, the resident macrophages of the brain. And I will put that in more of a neuro context, which is what we do. So because my lab in Paris really focuses on one big question, which is how the cerebral cortex, which controls sensory perception, motor behavior, cognitive function, how the cerebral cortex wires during early development. And so cortical wiring is actually a very long-lasting process that involves many sequences, starting in the embryo, early postnatal life, adolescence, adulthood. And this process is absolutely essential because alteration in any steps of these sequences can actually lead to neurodevelopmental disorders, which include autism spectrum disorder, intellectual disability, or even schizophrenia. And it all starts really very early in the embryo by a very complex choreography of cellular migration, assembly of circuits, and all this happening as the brain grows, folds, and changes of shape. And this early choreography is really controlled by genetic and epigenetic program, but there is also evidence that this process can be modified or altered by environmental perturbation. And this is really illustrated by the fact that, for instance, prenatal inflammation or inflammation occurring around birth is a risk factor for several of these neurodevelopmental disorders. And in my lab, really what we focus on is to try to entangle, understand, grasp how this complex cellular choreography leading to the building of circuits, how this choreography is regulated and coordinated. And we think it's going to be absolutely essential to really understand these steps, not only to understand how functional circuits emerge, but also, and importantly, how small deficits that can have either a genetic origin or an environmental origin, how this deficit can ultimately lead to pathology. And in this context, we study a variety of things, and one of these things is actually the role of microglia. So why do we focus on microglia and what are these cells? Microglia are the brain-resident macrophages, and as macrophages do, as tissue-resident macrophages do, they're really inside the tissue. They contribute to homeostasis, and they do that through a variety of functions as immune cells, mainly phagocyting, producing chemokines, cytokines, regulating the extracellular matrix, and so on. But a series of studies over the past, let's say, 15 years have really shown that these cells can do many more than their immune function. And I'm a bit ashamed to say that in this IMMUNOTALK. And this really has been started by the observation, by looking at these cells in vivo from different laboratories, showing that these cells here shown acting in response to a laser injury, but showing that these cells, even in the absence of injury, can actually have a very dynamic behavior and constantly scan their environment. And this has really led to the hypothesis that these cells could be doing much more than their immune function or potential immune function in the brain, but also contribute to the homeostasis and the development or maintenance of circuits. And a series of absolutely landmark studies over the years have shown that indeed, in the postnatal and adult brain, microglia have a key contribution to the development and maintenance of circuits. For instance, they're important to regulate neurogenesis, the survival of neurons, but also a key for regulating synapses formation, especially excitatory synapses, as well as their elimination or synaptic pruning. They're also very important to regulate myelin, which is a sheath around axons, which is important for regulating the transmission of electrical activity. And they're also very important to regulate neuronal excitability, and so forth. So microglia have really emerged over the past 15 years as key regulators in the postnatal and adult brain as actors in the physiology, basically, of cerebral circuits. So cells that can do many things in the postnatal and adult brain in physiological conditions. And in a way, illustrating or as a hallmark of this role, microglial dysfunction has now been involved in almost all the brain disease, from neurodevelopment to neurodegeneration. So microglia interact very closely with their neighbors, and they do that through a variety of signaling pathways and receptors. One of them is the CSF1 receptor pathway, which is absolutely required for the maintenance and survival of these cells, as for most macrophages. But it's also, they're also using a variety of signaling pathways to interact with their neighboring neurons. And just to name a few, CX3CR1 fractalkine, the complement pathway, purinergic receptors, or lipid sensing receptors that are actually key for the functions of these cells, again, in the postnatal and adult brain. And in agreement with the fact that these pathways are important, mutation in several of these pathways have been linked to disease. So cells that really interact with their close environment, with their niche, and cells, again, as macrophages, that are really, really sensitive to their environment and to environmental signals. And for instance, as a hallmark, of course, these cells are highly responsive to inflammation, but several lines of work have also shown in adults that they can really, really respond or be modified, for instance, for body-derived signals, such as the one from the microbiota, and for instance, short-chain fatty acids. So cells that do many things in the postnatal and adult brain, but that can also be, in a way, plugins into the brain to convey or to mediate the influence of body-derived signals. So as another hallmark of these cells being highly responsive or reactive to their environment, it's now well established, in particular through a series of single-cell transcriptomic studies, that microglia exist in a variety of states. So not substates, but really states. So in adults, especially in the adult brain, these cells tile the parenchyma, as you can see here, and they've been identified as being in a sort of homeostatic cell state, even if they constantly survey their environment. But it's an homeostatic state that has been defined in particular by transcriptomic signatures. And it's also known that these cells can engage or adopt a variety of different states, especially during aging or disease. And I will just name a few of these states, and one of them is the disease-associated microglia, which has been originally identified in Alzheimer's disease mouse models, but has now been found in several neurodegenerative conditions, which is also characterized by very specific transcriptomic signatures. Quite remarkably, these different microglial states have also been described, in particular in development, this time normal physiological development. And one of the states that has been well defined in the postnatal brains that I will be referring to later on is called axon tract associated microglia. It has also been named proliferative associated microglia or youth associated microglia. And it's microglia in these states accumulate in specific parts of the brain, specifically the postnatal white matter, which is where axonal tracts are formed, and these cells accumulate there. And the signature also that these cells have is very well described and actually shares some feature with this disease-associated microglia found in neurodegeneration. These postnatal ATM have really been involved in myelin formation and growth, and also the regulation of the cells that will produce the myelin. But for earlier steps of development, for instance, in the prenatal brain, we know that there is a wide variety or heterogeneity in microglial state that still remains largely to be characterized. So cells that have many functions in the postnatal, adult, and aging and diseased brain, cells that interact closely with their environment, that exist in response to this environment in different states, really raising the question of whether these specific states could be exerting specific or specialized function, but this really largely remains to be addressed. So why look at microglia in very early steps of development? We started really to focus on these cells as developmental neurobiologists when it was shown by Myriam Merad and Florent Ginoux that microglia actually originate outside of the brain in the yolk sac very early and colonize the brain at its earlier steps. In mouse, it's around E8.5, E9, embryonic day 8.59. In human, it's gestational week 4-5, so really early in pregnancy. And these cells seed the brain, and as a population, stay in the brain, self-renew, and expand. And for us, it had many important consideration or implication, because it means that these cells basically, at the first place, these cells are actually a main glial population in the brain very early on. Why? Because other glial cells, meaning non-neuronal cells, such as astrocytes or oligodendrocytes, are generated much later in the brain, but microglia as immigrants, early immigrants, arrive much earlier. And so that means that these cells could have very specific population and specific roles in the development of circuits. Anything that alters these cells could have lifelong impact on the brain. And finally, these cells could be very important or key recipient for external modulation, such as inflammation, in and mediating this effect in the developing brain. And so over the past 15 years, we've really focused on what these cells are doing at very early stages in the prenatal brain. And today, I will tell you a bit about the fact that these cells play very early and specific roles that diverge a bit from what they can do later on, that altering these cells can have lifelong impact on brain circuits, and that they convey also environmental modulation, being sensitive to these environmental modulations. And so I will mainly summarize some of our published results to really focus on one novel function that we've identified for microglia, which is to maintain or to preserve tissue integrity during the complex morphogenetic and morphogenesis and building of the brain. So really a structural role in preserving integrity. So what can we say about microglia in the embryo? So one specific feature of microglia that you can see here on this hem section of an embryonic brain at E14.5, using the beautiful CX301 GFE mouse line generated by Stefan Young, which really enables us to look at these cells. One specific feature is that in contrast to the adult or more postnatal brain, where these cells completely tile or invade the parenchyma, at these early stages, microglia have a very uneven and heterogeneous distribution, and you can appreciate with some accumulation and some zones that are devoid of microglia. So importantly, these microglia lie within the parenchyma of the brain, in contrast to what has been called border-associated microglia, or also CNS-associated microglia that really lie, for instance, here in the meninges and are not inside the brain parenchyma. And I'll be focusing really on microglia today. So these cells are have a highly heterogeneous distribution, and this distribution is not related, for instance, to cell death or elimination of dying cells, because there is very few apoptosis at this stage, but rather they're really related to events, developmental events occurring in the brain at this time. So for instance, in the cerebral cortex, in the neocortex, originally they're excluded from this structure and progressively colonize it. And I hope you can appreciate that by postnatal day 20, P20, these cells have acquired basically an adult distribution. So very progressive colonization. And these different accumulation are actually associated with specific events. For instance, here the formation of this boundary that I'll be mentioning again, or the formation of specific axonal tracts, or in the cortex, they're tightly related to specific cells that invade the brain that are called interneuron and that are important to modulate and regulate rhythm and activity in cerebral circuits. So cells that are really adopting very specific location interact with specific axonal tracts, streams of migrating neurons, and this distribution really raised the question of what they could be doing in the early brain at these early time points. And so for that, we really used a variety of approach, and I will summarize some of our findings. Where we really looked at what these cells do using several approaches that I'll be detailing after, mainly using mouse models that deplete microglia or mutations that alter microglia function. And over the years, what we could find is that really microglia are important for the positioning and distribution of the interneurons that I just mentioned, that are T cells important to regulate the activity of cerebral circuits. And most importantly, deficits in these interneurons have been linked as a causal factor, usually modified by genetic predisposition, but they've been identified as causal factor in the etiology of autism spectrum disorders or schizophrenia. We also found that they're important to regulate the progression of specific axonal tracts, for instance, dopaminergic axons, and that basically they exert this function through classical microglial pathways that have been linked to the regulation of excitatory synapses in the postnatal and adult brain, but here are modulating circuits at a different steps by regulating the development of these interneurons and formation of axonal tracts. We've also found that perturbing early microglia during the brain, or for instance, exposing embryos to prenatal inflammation, is actually sufficient to induce in the long term deficit in the wiring of these interneurons, which are going to be really important because they mediate inhibition in the circuits at later developmental stage and regulate activity of cortical networks. And that is associated to deficit, for instance, in sensory integration and in social interaction, which are core features of neurodevelopmental disorders. So in a way, microglia during the prenatal brain seem to be really important for the wiring of inhibitory interneuron, in contrast or in advance to a function that they have at later developmental stage in the wiring of excitatory circuits. And in this context, you will hear way more about what these microglial cells do in the postnatal brain by the fantastic Dory Schaefer, who will be talking in a nearby next week, I think, in the Global IMMUNOTALKs. We also found that actually microglia are highly sensitive to a variety of environmental signals beyond prenatal inflammation. And through collaborative studies with Florent Ginoux, we showed that microglia are really sensitive, even at prenatal stages, to the microbiota. And by analyzing germ-free mice and also mice exposed to antibiotics, we found that not only microglia can respond or sense or be modified by the microbiota, and during pregnancy, it's the maternal microbiota, but that also quite remarkably, these changes are really more specifically affecting microglia in males. And for us, it was really striking because in a way, all these findings combined really provided us with the idea that there is a true and important convergence of risks on the wiring of the specific interneuron. We know that genetic predisposition for these diseases can alter their development, but our findings really showed that in a way, potentially immune risks can converge on the wiring of the same cells. And in addition, these immune risks could be differentially implemented or exerted in the context of a male sexual identity, which is very important because we know that neurodevelopmental disorders have a much higher prevalence in males in the occurrence of these diseases. So real convergence of risks that we're now fully exploring. But today I will not talk about this, which is much more neuro, and focus more on another function that we found for prenatal microglia, which is to maintain, as I said, a tissue integrity. And this is really a work that was spearheaded by a very talented PhD student, Akinde Lawrence. So what did Akinde do? So we went back to look at microglia, and something that was very striking at prenatal stages is that not only these cells have a very heterogeneous distribution, but they also, at least morphologically, seem to be present in a variety of states. And for instance, we found that microglia could be really present in a very amoeboid-like shape, which is normally associated with a disease or disease condition, and in particular at the specific location here, which is a boundary between different structures, between the neocortex, the striatum, and the amygdala, an important structure for fear and anxiety. And we found that really here, or Akinde found that really at this specific location, these cells were present in a very odd morphology. They are bona fide microglia, as we can see from very specific markers such as P2Y12, but also from their fate map tracing. And cells at this corticostriatal amygdalar boundary or CSA boundary have this particular shape. And that raised the question of what could these cells be doing, and are they in a specific state, and do they have specific function at this position? And so the first thing that we did was to look at whether a microglia in the prenatal brain would indeed be existing in different states. And for that, we went back to single-cell transcriptomics and reanalyzed data sets in the embryonic mouse that were actually available. And by re-examining them, we found that microglia are present in a relatively homeostatic cell state, cycling state, which is in agreement with the fact that these cells can proliferate and colonize the brain. But we also found very strikingly a signature that was very, very close to these ATM states that was described postnatally and that I've been mentioning that are present in the postnatal white matter and important for myelin formation. And we could find that these cells have a very conserved signature, and in particular, we found very core, maintained, differentially expressed genes that are characteristic of the ATM signature. For instance, SPP1 or osteopontin, GPNMB, ITGAX or CD11C, CLEX7A, LGAS3, which encodes galectin-3 or MAG2. And so this really showed that we can find also ATM-like cells in the embryo. And we further found that indeed these cells are specifically present at this accumulation, this cortico-striato-amygdalar boundary that I will call CSA, to make it shorter, and that indeed 60% or more of these cells present here specifically co-express all these markers that are characteristic of the ATM profile. We also found that these cells are actually highly phagocytic, and they have not only this amoeboid morphology, but by using ex vivo phagocytic assays, we found that they can largely engulf a lot of material. So in the embryo, at the specific boundary between different structure, we find this microglia that are in a specific state. Importantly, a very similar or conserved accumulation is found in human embryos at the same location, really confirming that we can find at prenatal stages cells that share transcriptomic, cellular, and morphological properties of these postnatal ATM in the embryo. And we call these cells ATM-like or embryonic ATM, because of course they're found at a different time point. So this is showing that indeed, in contrast to the adult, where we have most cells in an homeostatic condition, even at very early stages, we find this sort of heterogeneity in cells, in states present at specific location in the fetal brain. So the next question was, what could microglia be doing at this specific spot? And for that, we turn to experimental models that we have that are all imperfect, but in combination enable us to address the question of microglial function in the embryonic cerebral tissue. So the first strategy is to actually deplete microglia, or rather I should say to deplete microglial progenitors before they enter the brain and assess the development of the brain in the context of the absence of microglia. And for that, we can target the CSF1 receptor pathway either by injecting very early blocking antibodies when cells are generated in the yolk sac at E6, E7 embryos. And as you can see by flow cytometry, this strategy leads to a very drastic depletion of microglia at embryonic stages. You still have a few of these cells that can enter the brain and stay there. And from these cells, the microglial pool really replenishes and repopulates the brain. And this is largely done by P7, by postnatal day 7, as you can see here. We can have exactly the same depletion model using a pharmacological inhibitor of CSF1R, PLX3397, that we can use also from early stages. And I hope you can appreciate that this depletion is very drastic, as you can see here by immunostaining. This is at E14.5 and this is at E18.5, where we mostly lack microglia in the brain parenchyma. We can combine this with a variety of other models, a very ugly one, which is P1 mutant that blocks the development of all myeloid cells. And we can also use a model that has been developed by David Hume and Claire Prudence, which is called the CSF1 receptor fire mice, which lacks a specific regulatory element in the CSF1 receptor gene, and which selectively impairs resident macrophages without drastically impairing other macrophages. And in the brain, it really enables the selective depletion of microglia versus border-associated microglia that are largely preserved. And so this model really has been important to tackle microglia-specific functions in the brain, especially very recently by a beautiful study by Véronique Miron. We can also look at other mouse mutants that affect the functions of microglia in the postnatal and adult brain, and that have been widely used to look at excitatory synapse remodeling, especially again during postnatal development, which target the CX3CR1 receptor pathway, tyrobp TREM2 or complement receptor pathway. And we can also do prenatal inflammation, for instance, by exposing pregnant dams to either a lipopolysaccharide or poly-IC that respectively mimic bacterial or viral maternal infections. And so using combination of these models, we can very broadly affect microglia at early stages and look at the consequences on brain development. And so one question was really what happens at this specific boundary that we've identified here at this CSA. And our findings were relatively surprising, or at least to us, we were not expecting that. What we found is that when microglia are gone, at this border, the two edges of the boundary detach, and we have now the presence or the formation of a cavity at this spot. I should stress here that this region is a border between structures that normally grow in very different directions and paces, namely the cortex and the striatum and the amygdala, and it's a known site that has been described by embryologists. It's a known site of intense morphogenetic constraints as the brain grows. And so what we see here is that when microglia are lacking at this spot where they normally accumulate, we now see a detachment and a formation of what we call the cavitary lesion. This was absolutely not observed in any of the models that we know or are important to regulate microglial function in the postnatal brain or in mild maternal immune activation prenatal inflammation, showing that dysfunction of microglia is actually probably exerted through different pathways. Importantly, this lack of adhesion formation of this cavitary lesion is sort of amplified, or it's still detected at later stages at the end of embryogenesis. I hope you can appreciate that this cavitary lesion is maintained or rather enhanced as developmental proceeds. And to really confirm that this is actually due to the lack or the absence of microglial function, we can do short-term depletion just around the time of the accumulation by doing targeted PLX exposure, or we can look in the CSF1 fire mice, and we find very similar phenotypes, really showing that this is due to the activity of microglia in the prenatal brain. So this was quite surprising. Microglia are important to maintain really the integrity of the tissue of this boundary at this site of really high tension and morphogenetic constraints between structures that grow in different directions and at different paces. And because sectioning can lead to deterioration of tissues, and we wanted to make sure that this is not an artifact, but this is a real function of these cells in the embryonic brain, we turned to transporization, tissue clearing, and also MRI scans that enable to address or visualize the brain tissue or brain parenchyma in the intact brain. So tissue clearing really showed, and this is a lateral view of the brain, that we can detect the presence of this cavity in all the models when we lack microglia. And I hope you can appreciate in 3D that this is really a relatively large lesion, but that has a sort of tear shape to really localize in the temporal lobe, in the caudal part of the brain, at the border again between the entorhinal cortex, insular cortex, the striatum, and the amygdala. And by doing MRI scan, and this is in collaboration with Luisa Ciobanu in Neurospin Saclay, we showed also that in the intact brain, we don't have any appearance of lesion in this spot, but either in PLX or in fire mice, we still have very clearly this hyperintense signal in MRI showing these lesions located at this spot at mid-neurogenesis or at the end of embryogenesis or in POPs. So this is really showing that microglia at these specific spots of accumulation where the cells are in a very specific state, ATM-like state, when the cells are absent, we have the formation of cavitary lesions, showing that microglia are important to maintain tissue integrity and the normal state of the tissue during embryogenesis. So the next question was, can we find potential pathways that could be important for this microglial function? And as I mentioned, all the classical microglial pathways that are normally associated with their role in the postnatal or adult brain didn't show any of these lesions. So, oh, sorry, I forgot that. So the question before that, the question is, can we find other regions where microglia would accumulate, where these cells would be in a specific state, and where their absence would lead to the formation of cavitary lesion? And for that, we did a very longitudinal study throughout development, and we could find only one other spot than the CSA where we had exactly the same situation. And this is another point of attachment of the cerebral cortex, this time not to the lateral part, caudolateral part, but here with the septum at the midline, just below a big axonal tract that enables the connection between the two hemispheres, which is called the corpus callosum. And here we have also, here at late embryogenesis, we have this accumulation of microglia, and we have also these cells present or expressing these very specific ATM markers, CLEC7A, SPP1 osteopontin, and so forth. And when we look in models that lack microglia, we also observe these cavitary lesions that are present here at the midline and that always are detected. So to come back to the question of the potential molecular pathways involved in this unusual and surprising role of microglia, we focused on ATM factors that are expressed by these cells, and we took advantage of full knockouts because at these specific time points, the only cells in the brain that express these genes are actually microglia. And we screened through several mutants, but we focused on SPP1, which codes for osteopontin, which is a cytokine that has been involved in many different functions, important for regulating or organizing the extracellular matrix, but been shown to be important for skin healing, calcification, response to inflammation, and many, many other functions. But what we found is that actually in SPP1 mutant, we partially recapitulate the phenotype that we observe when in the full absence of microglia, as you can see here, at the CSA and also at the midline. And we have in about 60 to 70% of the embryo that show similar lesions. Also, these resorb much faster, showing that SPP1 can contribute to the function of microglia in these spots, but certainly other factors that remain to be investigated are also contributing. Nonetheless, our findings really show quite surprisingly that in addition to a lot of the functions of microglia in really shaping circuits that use pathways also important postnatally, but to regulate the formation of other structure, we find that microglia, in part potentially involving SPP1 osteopontin, microglia accumulate in specific spots, and at these specific spots, they seem to be very important to maintain the integrity of tissue as the brain develops and grows. So without microglia, the brain develops with holes in the brain. So how does that work? And what could be happening in this specific region? And that has taken us a lot of time and thinking to really understand or to start grasping what could be happening there. And basically the light came by looking at what happens in physiological conditions at these spots. And we focused on the CSA. So as I mentioned, this is a region of tension that has been well described by neuroanatomists and developmental biologists between structures that grow in different directions. And this is also the case for the midline. And what we found is that actually at this spot, we have a sort of microcavities that are formed, and we see that ATMs start surrounding this region, appear around this region. And these regions actually comprise some microcavities that really resemble microlesions that are full of small cell debris. There is no massive cell death at these spots, but really they look like micro tearing in the tissue. And that really raised the question for us, or prompted the hypothesis that maybe morphogenetic tension in these spots that could alter the tissue could be important to recruit microglia, and also, in a way, could be important or constitute sites of vulnerability in response to morphogenesis and morphogenetic stress. So how can you test that? It's actually not very easy. So the way we tackle this was to use models in which morphogenetic tension would be really increased. And one of these models is a model of periventricular nodular heterotopia, where we have a huge accumulation mispositioning of cortical neurons that really changes the shape and the way the cortex develop and enhances tension throughout this structure. This model was developed by Silvia Capello and Magdalena Goetz. And what we found is that when in this model, when we increase tension, what we have is that we have tearing and the formation of cavitary lesion exactly at the CSA. And this occurs at the end of embryogenesis and also postnatal stages. So can this increase in tension can recruit microglia or change microglial composition? And we looked at this, and indeed we found before tissue tearing that the increasing tension can actually largely recruit microglia. And this is a recruitment because the total number doesn't change, but we have more cells present in this location. And moreover, we find that the microglia that are recruited are in a specific ATM state, for instance, as illustrated by the expression of the core factor important for maintaining tissue integrity, SPP1. So increasing tension leads to a break at the CSA, where we normally have microcavities, but it can also lead to the recruitment of microglia and the local induction of the specific ATM-like state. Conversely, what happens when we decrease tension? And we can do that by using another model that we developed several years ago that lacks a large structure of the brain, the thalamus. So it's a more caudal region, but it can change or allow some subtle changes in the morphology of the brain, and it decreases overall morphogenetic structure tension as the brain grows. And in this model, what we also found is that conversely, we have less recruitment of microglia, or at least less cells that are in an ATM-like state as assessed by SPP1 expression. So when we decrease morphogenetic tension, we decrease the recruitment of ATM-like microglia there. And when we deplete microglia in this context, instead of having lesions, now we have very small or no lesions at all, showing that microglia in a context of reduced morphogenetic tension are not so much required to preserve integrity. So microglia really protect the CSA from this morphogenetic tension and the formation of the lesions. So collectively, the idea, or what our findings show, is that morphogenetic tension at these structures actually recruit microglia and contribute to the induction of the state, and raising the idea or the hypothesis that these cells could be important to promote repair at these sites where we have microlesions and when we can have breakage or tearing of the tissue. And to test that, we first examined whether mechanical damage could actually, in utero, trigger the accumulation of these cells. And we did that by performing in utero poking using a glass pipette during a stab wound model. And you can appreciate that only within 2 hours and a half, so very short time period, that enables the recruitment of microglia that actually express, again, SPP1 and other core ATM markers. And during this time window, the presence of microglia is important to maintain a proper repair of the tissue, as you can see here. So mechanical damage can recruit these cells and induce this state. Are microglia or important, or SPP1 important for the repair of lesions? So to tackle that, we wanted to look at larger lesions, and we took advantage of the fact that in our depletion model, microglia can repopulate and come back. So what happens to the lesions in this context, and is SPP1 important for the repair of these large lesions? And this repopulation occurs postnatally, so we can have these lesions here. And I hope you can appreciate that when microglia repopulate, they accumulate at this spot, and the closure of the lesion is very rapid, starts around P3, so 3 days, and is mostly done by P7 when microglia is fully repopulated. Importantly, the accumulation of cells at the scar or the closure is always accompanied with a very high and local expression, again, of ATM core factors. And SPP1 is absolutely required for the proper closure of these lesions, as seen when we perform these experiments in an SPP1 mutant context. So I hope I've convinced you that in addition to their role in release circuits and regulation of synapses and integration of specific neuronal cells, microglia have a more fundamental and very basic function in preserving tissue integrity at specific spots where these cells are in a specific state, and that this function relies partially on SPP1 osteopontin, so different from these others function in the embryonic brain, and also relies on a very important capacity to repair, which is a core feature of many macrophages in very different tissue. And for us, this has really broad implications for our understanding of morphogenesis, because we think that this is showing a novel role of microglia, a very basic one, but in accompanying the proper morphogenesis of a brain and potentially a folding and changing of shape and the development of a proper cortex. For evolution, because these functions have not been identified, for instance, in zebrafish that have a much smaller brain with less tension. And so we think that this is probably a co-optation of an immune cell in the building of a complex brain, and the neocortex is a hallmark of mammals, potentially showing a sort of co-evolution between resident macrophages and morphogenesis of a complex mammalian brain that can also be important for more twisted or complex morphogenesis in other mammals, such as humans, but also for pathologies, because damage or hole or cysts in the brain are usually associated with the fact that microglia could attack the tissue. And here we find that a loss of function of this cell could actually lead to very similar lesions, and very similar cysts to what we have observed have been reported in several congenital infections or deficits in leukoencephalopathy. And we're actively looking at whether loss of function or perturbation of microglia could be involved in this process. So I hope I've convinced you that it's really important to look at what these cells do very early on, because that can have lasting consequences that are not necessarily obvious when you look at later functions. And I just want to stress that indeed we find that having these holes in this region, especially in the amygdala, has actually potentially long-lasting consequences also in the wiring of these regions. Having holes at specific time points in the brain can impact the way the brain wires later on. So I want to finish with the most important slide, which is to acknowledge the people who have done the work. So this was really spearheaded by Kinde Laurence, as I mentioned, but it's a team effort. And Cécile Bridlance, Alice Kanzi, Ludmila Lokman, Morgan Thion all contributed to this study. We have very key and important collaborators with whom we couldn't be working: Florent Ginoux, Luisa Ciobanu, Antoine Gello, Paolo Giacoboni, Laura Contini, and many more. And I also want to thank Fundings, and I want to thank you for your attention for this seminar. It's really been an honor and a pleasure to be here.
Sonia, the honor and pleasure is ours. It was a fantastic seminar. I learned quite a bit. I appreciate the fact that you dumbed it down for immunologists. So I totally think I learned quite a bit, and I think a lot of people will appreciate the language and the clarity you used in your presentation. As you can see here in this last slide, to those who have been listening or those who will be watching your seminar on YouTube, you can ask Sonia questions, and the way to do it is you go to, as you can see here on this slide, it's on Twitter. So Sonia, over the next 2 to 3 weeks, will be answering all your questions that you post to her on Twitter. She promised you take care of that. And so please ask Sonia anything you want to know. And as is slightly customary in our thing, we have a slightly abrupt ending. Not slightly customary, it's customary, but we have a slightly abrupt ending now. And I want to thank Sonia once again. I want to thank Global IMMUNOTALK for taking care of this incredible venue, and all of you for coming and listening to us. And we'll see you all next week. Thank you very much, and have a good morning, afternoon, evening. Bye-bye.